What do our cells look like? In 206 and 207, we're introduced to many complex factors such as DNA, proteins, other molecules, and some very complex molecular pathways. But what's interesting is how do these pathways and molecules interact? Are they occurring all at the same time? Where are they occurring in the cell? Kind of giving us a picture of what does the cell look like. And this is a very complicated question, and so let's start with the nucleus. This is what I'm calling nuclear organization. It's really a question of how do these processes organize? Where are all the molecules? How and when do they occur? So let's ask some questions. What does the inside of a nucleus look like? Specifically, given a cell and interphase, is the DNA in chromatin or chromosome state? Is the DNA randomly intertwined, or maybe are each chromosome separated from each other? Is it moving, or does it maybe stay in the exact same position for the cell's entire life? So these are some questions that can maybe give us a hint of what the inside of a nucleus looks like. Let's begin with the concept of chromosome territories. Inside a cell nucleus, we can see that DNA is in a chromatin state. However, it occupies a very distinct area within the nucleus. This is called a chromosome territory. You can see that they are distinct from each other with small little intermingling spaces. This is visualized here experimentally as each chromosome is fluorescing with its own particular color. So you can see that they are in fact distinct from each other. All of this is to say that the arrangement of chromatin in the nucleus is highly organized, and this high degree of organization permits and enhances some processes, some important processes that occur in the nucleus. If we're zooming in, we can see 3 chromosome territories here, all of the DNA separated from each other, but with strands of chromatin interacting and intermingling in these areas here. According to our legend, we can see that this is RNA polymerase, that these black strands are mRNA transcripts, and these are the chromatin loops that are being transcribed. And so the phenomenon that we're seeing here is active genes clumping together, being transcribed in these transcription foci. The formal term for this is a transcription factory. So generally, transcription in the nucleus occurs in these transcription factories where transcription machinery is actually shared between genes that are on the same chromosome and between chromosomes. So in these intermingling spaces is where the transcription occurs. There are many different kinds of transcription factories with all different kinds of properties. This is the general concept that genes that are actively being transcribed localize and group and are transcribed. But different kinds of transcription factories can have different properties. And so we'll highlight one type of transcription factory of importance, the nucleolus. It's a transcription factory for rRNA, or ribosomal RNA. You may recall from high school that the nucleolus is where ribosomes are created. And now we know that it's a ribosomal transcription factory. And so the defining features of this particular type of transcription factory are, 1, the high levels of ribosomal transcription machinery, specifically RNA polymerase 1. And the chromatin regions that associate are those that contain the ribosomal genes. These vary, of course, from species to species, but they'll consistently be the genes associated with ribosome production. And interestingly, we can actually have 1 to 4 nucleoluses per nucleus, depending on the species and the cell type.